All right, it's 12:30. And I'm happy to see you after a long time.
Yeah, good afternoon.
You all survived test 1. We'll talk about that later today. I hope we have time for that. But first, I want to make a couple of announcements. One is I received a notice that from now until December 4th is the period when students are encouraged to evaluate their courses. And you should be receiving, every time you log into Canvas, you should be receiving an alert asking you to review the course. I would suggest you don't do that until later in the semester, because you have until December. And if you wait, you'll have a better sense of what the course is all about. So you can evaluate it more thoroughly and realistically if you wait a bit. But anyway, if you see these alerts, yes, was there a question?
Oh, sorry. I was just gonna say, like, I'm pretty sure we get multiple evaluations. Like, there's one at the beginning of the semester, there's one and there's one at the end of the semester.
Oh, okay. Well, I see. So, have you been receiving notices?
Yeah, I got an email about it.
Okay. All right. Well, then what I'm saying is redundant with what you already know. And so I won't make any more issue about it. The other thing I want to announce, you've probably already seen it, and that is I issued an assignment. And this is a trial run to see if we can get a more thorough understanding of the material that we go over in this course. So what I'm going to do is go over the slide set for today. And then after the period is over and after the recording is available, I would like each of you to submit at least one issue, a question or a comment about the slide set. And this will be due on Saturday, so that I'll have time to go over your responses and organize them, and we can discuss them then on Monday. So this is an attempt to embellish and enrich the content of the course. So we'll see how you like this. See if you think it's helpful. If it's not, we'll do something else. But it's a first attempt. So you do have that assignment. It is posted. It'll become available after class today. Now, what I'd like to do first of all is go over the next slide set, which is a very important one, on mutations. Mutations and the CFTR protein. Mutations are especially relevant to this protein because this is the basis for your special project that we'll talk about later. And each of you will have a mutation of this protein assigned, and you'll do your report on your particular mutation. So CFTR stands for the cystic fibrosis transmembrane conductance regulator. So what about it? It's encoded by a particular gene. Where is that gene located? We went over this in a previous lecture. The location code is 7q3-2.1. That means the 7th chromosome, the Q arm of the chromosome, which is the long arm, section 3, band 2, subband 1. Now, how many nucleotides make up this gene? In other words, how big is it? Well, it's very large, over 250,000 nucleotides. And those encode for 1,480 amino acids. Now that's a lot more base pairs than what's needed to code for 1,480 amino acids. And how can we explain this? We explain it, and I will explain it based on the fact that this gene and many genes in eukaryotes contain introns. I'll tell you what an intron is. And the function of the CFTR protein is to transport chloride ions across the membrane from the interior of the cell to the exterior. Introns. So the wide gray bars here are what's called exons. That means it's regions of DNA that encode a protein. And the thin gray bars are introns. That means they're in between the exons and they do not code for proteins. Sometimes these sections are referred to as junk DNA. I don't think that much of it really is not junk. It has a purpose. But anyway, the CFTR gene has 27 exons and 26 introns. There will always be one more exon than there are introns in any gene. And this brings us to splicing. How do we deal with introns? Splicing to get a messenger RNA, mRNA. So if you go to the left here and you see a stretch of DNA, and the dark blue bands are exons, the pink bands are introns. They alternate. When that is transcribed into RNA, it's called a pre-RNA because both the introns and the exons are transcribed. However, after this transcription, splicing occurs so that the introns are spliced out, the ends of the exons are joined, and so now we have a mature messenger RNA. This is the one that gets read, translated into protein. This is splicing. So the protein then that results, you see here, that's the primary structure, which then, as you know from last time, gets folded and coiled and bonded by several different kinds of bonds into a tertiary structure, which has activity. At the lower left, I've circled something that's important. You should remember this. And that is that introns and intron splicing occur only in eukaryotes. Prokaryotes do not have introns. And the splicing occurs in the nucleus. It's only after the splicing occurs that the mRNA leaves the nucleus and goes into the cytoplasm. So you're probably wondering, why are there introns? And by and large, we don't know. We don't know why eukaryotes have them and prokaryotes don't, but we do know a few things. So for example, some introns encode what are called ribozymes. Ribozyme stands for ribonucleic acid enzyme, and it's a form of RNA that has catalytic activity. So you know that enzymes are catalysts. RNA has many functions, one of which is that it also can catalyze reactions. And in the illustration to the right, you can see how ribozymes can operate by cutting their own messages. So what they can do is catalyze the hydrolysis of their own phosphodiester bonds, the bonds that hold the RNA chain together. These can be cleaved so that the chain breaks. And you see that illustrated here at the bottom. And usually, for any particular message, the ribozyme will cut it always at the same place. So now you've known about 3 types of RNA: mRNA, tRNA, and rRNA. Now you can add to that ribozymes, ribozymal RNA, which is distinguished by the fact that it's none of the first 3, and it has catalytic activity. So any questions about introns or their functions? This shows how ribozymes can be involved in the regulation of gene expression. So if there is a message, an mRNA that encodes a ribozyme and it's near a gene, if the ribozyme is inhibited by a small molecule, it leaves the RNA alone and the gene is expressed. If, however, the ribozyme is not blocked, it will cut the mRNA and the gene does not function. So it's like a switch. If the ribozyme is there, it destroys its own RNA and the gene does not function. If something blocks it, it functions and the gene is expressed.
I have a quick question.
Yeah, your name?
Yusuf.
But, okay,
Yeah, just to make sure, you said there's always one more extron than an intron?
yeah.
Okay. Do you know why that is, or does it not
Well,
matter?
It does matter.
Okay.
And the reason is that introns are always between exons. So if we go back to that picture, you see introns, the thin gray lines are always between exons. And so you never have an intron outside a gene, which means that the end of both ends of any given gene will be exons. And that means there will always be one more exon than there are introns.
And just to clarify, the extrons are the bigger blocks, like 1, 2, 3.
Just a minute, I want to note who spoke. That was Emilio. Say that again.
So the extrons are like the 1, the 2, the 3, not the little lines in between, which are the introns, correct?
Right.
Okay.
The fat gray bars are the exons.
Yeah, thank you. I'm just trying to visualize it.
Yeah, it's always good to visualize. It helps you get the concept. Okay. Now, I just said that introns can be spliced like this. In this case, all the introns were spliced out, and you end up with a message of pure exons. That doesn't necessarily have to be the case. So here we have several examples of alternative splicing. So at the top, number 1 is full splicing. So there are 3 exons here, in red, blue, and green, separated by 2 introns. Well, if both those introns are spliced out, you end up with an mRNA genes 1, 2, and 3. Now, if there is exon skipping, that means instead of cleaving at both ends of the intron, you cleave at just one, then the entire, both introns and exon 2 are eliminated, and you end up with only exons 1 and 3 join. Or you can have alternative, either alternative 5' or alternative 3' splice site selection. In either case, you end up with a truncated exon 2. In one case, it's one end that's lost, the left end, and in the other case, it's the right-hand end that's lost. Or you can have intron retention. That is, one intron, the first one can be spliced out. I should say the second one can be spliced out. The first one left in which case you end up with an mRNA that has all 3 exons plus intron number 1.
Question, Professor, as Steve Stinson.
Okay.
If it's truncated, does that mean that it's flawed or inferior, or it's just a way that it's going to be read for that purpose?
Yeah, the latter. It's if it's truncated, all these alternative ways are actually ways of regulating gene expression.
Oh, okay.
They're always functional.
Okay.
So I have
Right.
here at the top that alternative splicing is a way to regulate gene expression, and it's a way to introduce variation. Later on, we're going to talk about evolution and natural selection. And so this is one source of variation for that.
Okay.
So let's take an overall look at the CFTR protein. So that's what we're talking about. Here you see a diagram of it. You can see that it's embedded in a phospholipid bilayer, a membrane. So the inside of the cell is at the bottom, the outside of the cell is at the top. And the whole point is that chloride ions have to get across this membrane. And as we noted earlier, phospholipid bilayers are very difficult for anything to cross. And so they need help. And in the case of chloride ions, that help comes from the CFTR protein. So you can see that there are what are called transmembrane domains, TMD1 and TMD2. These are the regions, so the protein is folded back and forth on itself. And the 2 domains are inserted in the membrane such that they go all the way across and form a channel. It's like a little tube through which chloride ions can be transported. Now, there are 2 cytoplasmic nucleotide binding domains, NBD2 and NBD1. These provide the energy. So chloride ions are actively transported. They require energy. Each ion needs a tiny bit of energy to get through this transporting protein. And that energy comes from the hydrolysis of ATP, like this. So within the nucleotide binding domains, they're there for the generation of energy to power the transport of the chloride ions. And it involves ATP. You see the structure of it here. The critical feature here, and you know that the adenine nucleotide is part of the structure of DNA. Well, this has nothing to do with DNA. This has to do with providing energy. So this triphosphate on the side of ATP, the terminal bond, this PO bond, is high energy. So that if it is cleaved off with the addition of water, you get a phosphate ion and ADP, and energy is released. And that bit of energy is what's used to transport chloride ions. And I put this in just to help you visualize that ATP and ADP are constantly interconverting from one to the other and back again. So it's a cycle. ATP cleaves its terminal phosphate, releases energy for work. The ADP then takes that phosphate, takes energy from somewhere and makes ATP. And this keeps going around and around. Energy is released, energy is consumed. Where does the energy come from? Well, for us, it comes from the food we eat. Now, I want to go over this diagram, but first I want to show you a video which I hope will help to clarify what's happening here. We're going from the nucleus, from the gene encoded in a chromosome, to a protein out here embedded in the membrane. And it's a very complicated process going from the nucleus to the membrane. And I want to now stop sharing my screen and show you a video.
Pharmacology of transporting epithelial playlist. And this video, what we're going to study is we're going to study a cystic fibrosis transmembrane conductance regulator, which is quite a complicated anion channel. So the cystic fibrosis transmembrane conductance regulator. So it's got a massive, great name, and it's often usually abbreviated as CFTR. I don't know what happened to the conductance. And so some people often just refer to CFTR as the cystic fibrosis transmembrane regulator, but its full name is the cystic fibrosis transmembrane conductance regulator. And basically what it is it's an anion channel, a channel which allows anions, which are negatively charged ions, to pass through it. Now, one of the main ions which passes through it is chloride. Chloride has a negative charge, so it's an anion, but there are others that also pass through it. Now, in this video, what we're going to do is we're firstly going to study the structure of the cystic fibrosis transmembrane conductance regulator, and then we're going to study how you actually open it, because it is not just a channel that is open all the time. There is a series of events that cause it to open, and they're quite complicated. It is not a simple channel by any stretch of the imagination. So we'll begin with its structure. So basically, it consists of two transmembrane domains, which both have 6 spanning alpha helices, making them up. Okay, so you start off down here, basically, with the amino terminus, and it is one polypeptide that makes up the entire cystic fibrosis transmembrane regulator. I missed out conductance now. The cystic fibrosis transmembrane conductance regulator. So you start off with the amino terminus of the polypeptide down here, and then it has an alpha helix which spans the membrane, and then it has another alpha helix which spans the membrane, and it continues on, and it does it 6 times. So like that. So it ends up with 6 alpha helices which span the membrane, and that is known as one transmembrane domain. So I'll call this transmembrane domain 1. Okay, all of that. All of those 6 alpha helices which span the membrane are known as transmembrane domain 1, or TMD1 for short. And the individual alpha helices, if you want to label the individual alpha helices which span the membrane, you label them T1, then T2, T3, T4, T5, and then finally obviously T6 here. So basically, the transmembrane domain 1, TMD1, consists of 6 membrane-spanning alpha helices labeled T1 through T6. Then what you have is a loop connecting this to a second transmembrane domain with 6 alpha helices spanning the membrane. But there are some important domains in this loop. There is what is known as the nucleotide binding domain 1. So I'll put this here, nucleotide binding domain 1, okay? Binding domain 1. And I should have drawn that with 2 sort of binding sockets there, so I would like another indentation there. Okay, nucleotide binding domain 1, and obviously that's also abbreviated often to NBD1. Okay, and then you have a second important domain in this loop connecting the transmembrane domain 1 to the transmembrane domain 2, which is the R domain, or the regulatory domain. So this is the regulatory domain. Regulatory domain.
I'm going to start this over.
Okay, so welcome to this next video in pharmacology of transporting epithelial playlist. And this video, what we're going to study is we're going to study video in pharmacology of.
This is not the video I wanted, so let me go back and try to get the one I wanted.
Okay, so welcome to this next video on CFTR. And in this video, what we want to discuss is the synthesis of CFTR and how we traffic it to the apical membrane. And particularly what we want to focus in on is how a very common mutation in cystic fibrosis, namely the delta F508 mutation, can disturb this process. Okay, so firstly, let's begin with the cystic fibrosis transmembrane regulator gene, which is on chromosome 7 in the nucleus. So if we have some cell here, so here's our epithelial cell, then in the nucleus, what we have is we have 23 pairs of chromosomes. Now, on chromosome 7 is where you will find the CFTR.
Can you see this and hear it?
Yeah.
Yeah,
You can?
Yes, we can.
Yes.
Okay, all right, then we'll see.
Gotcha. So let's take A, chromosome 7. So what we have here is chromosomes have 2 arms, and one is a short arm, this top one here. So this is the short arm of the chromosome. and the other is the long arm. Now, the short arm is referred to as the P arm, and the long arm is referred to as the Q arm. So this is the P arm, and this is the Q arm. Right, and this is chromosome 7 that we're talking about. Now, somewhere in chromosome 7, you will find the CFTR gene. So let's pull it out and imagine we've got it over here. So we've got this strip of DNA here, which is going to represent our CFTR gene. So I'll just complete the double helix. Okay, so let's imagine that, where's my highlighter here? Let's imagine that this represents the whole CFTR gene. So all of this is the CFTR gene. Now, the CFTR gene is a very, very big stretch of DNA. It consists of 250,000 base pairs. So basically, if you count the number of organic bases that are paired in this strip of DNA, and you go all the way along from the start of the CFTR gene to the end of the CFTR gene, you will count around 250,000 of them. So that is a big strip of DNA. Now, the protein is not coded for by a gene that size. Most of it is non-coding. So most of it is what are known as introns. So basically, there are strips of this DNA that are actually going to be used to create the CFTR protein, and those are referred to as introns. So let me draw them on. So basically what we're going to have is little strips of this that are actually used for coding and used to actually make the CFTR protein itself. And all the bits in between are just junk, basically. They're not going to code for anything. So these are known as exons. So between these two lines is an exon, a bit of the DNA which we're actually going to use to construct something. And basically in the whole CFTR gene, in this whole 250,000 base pairs, there are 24 exons. And all the bits in between, so these big bits here, these are introns or non-coding DNA. Right. So when we transcribe this huge, great CFTR gene into mRNA, what we're going to do is we're going to transcribe all 250,000 base pairs, and we'll end up with a strip of mRNA like this. And if I mark on the exons on here again, so here are these 6 exons that I've drawn. Of course, in reality, there'll be 24 of them. So these are the exons. These are the bits we actually want to use to make our CFTR protein. And all the bits in between are introns. So this is an intron and this is an exon. Right. And this overall is a piece of mRNA. But this is actually what is known as pre-mRNA, because it's the bit that we just literally transcribed from the DNA. Before we're actually going to make it, before we're actually going to use it to make protein I, before we're actually going to put it through a ribosome and let translation occur, what has to happen is you have to cut out all of these introns, these bits that aren't going to code, and just stick together the exons basically to make what is known as a mature piece of mRNA. And that process is known as splicing. So this is the process of splicing. So basically what we're going to do is we're going to take this intron here, the start of this exon, we're going to get rid of the introns. So this is this first bit of DNA of RNA that we actually want to keep. Then we're going to take the second bit from here. Then we're going to, so I'll just mark this. Then we're going to take the third bit from over there, we'll end up with the fourth bit from here, fifth bit on, sixth bit on. So basically we're going to cut out all the clutter and just keep the coding bits and then join them all together. And then what we get is a mature piece of mRNA. This then goes on to the endoplasmic reticulum, which is a membrane-bound structure which is very closely associated with the nucleus of the cell. So this mRNA is going to go up into this endoplasmic reticulum, and it's going to be translated in this endoplasmic reticulum. So I'll draw this piece of mRNA here. So it's just a strip like that. And basically what is going to happen is that you're going to have a ribosome here, which is going to use this mRNA to make you your CFTR protein. So you end up with your CFTR protein. So I'll draw my CFTR protein here, okay? So this is the CFTR protein. Right, and now what I want to discuss is what is going to happen to the CFTR protein next, basically. So next what happens is that in the endoplasmic reticulum, the CFTR has what is known as core sugars added onto it. So basically, what is meant by a core sugar is, so if I do an example down here, if you remember from biochemistry what a glucose molecule looks like, it's basically this hexagon molecule. And you'll remember that if you make starch or glycogen out of glucose, what you effectively do is you bind glucose to glucose over and over again. And you bind the hydroxyl group on this first carbon, because this is actually an oxygen over here. So this is counted as the first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and the sixth carbon actually sticks off of there. Basically, you bind the hydroxyl group of the first carbon to the hydroxyl group of the fourth carbon, and you create these long repeating polymers, basically structures. And that long repeating structure, if you just continue on like this, that is what is known as a core sugar. It's basically monosaccharides, which is just a single sugar molecule like glucose, fructose, galactose, bonded together in this predictable manner like this forever. Now, there's another concept that we need, which is a complex carbohydrate or a complex sugar. And basically, complex sugars are when you mess it up, when instead of just doing the linear pattern, so let's do a bit of the linear pattern, but then you also have other sugars bonded onto the different hydroxyl groups. So if we just draw this one here, basically this sixth carbon up here also has a hydroxyl group, so you could make a similar bond like this, but that messes up the pattern and you get much more complicated sugars, and that's what's known as a complex sugar, where you have basically new sugars linked to the old ones of the carbohydrate chain, but you don't link them in this simple repeating pattern from the first hydroxyl group to the fourth hydroxyl group. Instead, you start linking them off of these other chains, basically, and then you get a complex sugar. So basically, in the endoplasmic reticulum, simple core sugars are added to the structure of CFTR. So CFTR at the moment is just pure protein, it's amino acids, but you're going to, it's a more complicated structure than that. So you're going to add on sugars to modify structure, basically. And in the endoplasmic reticulum, the type of sugars that you add on are core sugars. So you add on the core sugars. Then what happens is you traffic the CFTR to what's known as the Golgi apparatus. So here is my best picture of the Golgi apparatus. So the Golgi is a system of interlinked membrane-bound structures. And basically what's going to happen is that CFTR is bound in the membrane of this endoplasmic reticulum. The endoplasmic reticulum is going to split off vesicles, effectively, with the CFTR in. And these vesicles, CFTR, and these vesicles containing the CFTR are going to deliver it to the Golgi apparatus. Okay, so once it gets to the Golgi, so let's draw it in the Golgi now. So let's draw CFTR over here. Once it gets into the Golgi, it undergoes further modification. So you start adding complex sugars to it. So in the endoplasmic reticulum, you add core sugars, and in the Golgi, you add complex carbohydrates. So I drew a simple sugar there, so I'll draw that same simple sugar. So maybe what happens is the Golgi apparatus starts modifying the core carbohydrate so to turn it into a complex carbohydrate. So let's draw some bits coming off like this, which is where you've attached these new sugars basically onto usually the 6' hydroxyl group. Okay, and then finally what happens is the completed CFTR goes into another vesicle and is trafficked to the apical membrane of the cell. And that's all good. So here's the CFTR going off the apical membrane of the cell. There is also a process for taking it out of the apical membrane of the cell. So once it does get to the apical membrane of the cell, you need another process for removing it from there, should you decide that there is too much of it. And the process is basically clathrin-mediated endocytosis. So here is our CFTR happily up in the membrane now. And basically another process can remove it and return it back into a vesicle, which is clathrin-mediated endocytosis. And these vesicles can then, these endosomes, as they're called then, once you've
Professor, can you hear me?
Yeah,
Okay, I have a question. Maybe I missed this in the beginning of it, but is the CFTR happening from when the mRNA is replicated from the DNA? So it's happening in the strand that comes off the DNA?
okay, let me share my screen with you again. Can you see that?
Yes.
Okay, you can. All right. Okay, now we're back to the slides. Can you repeat your question, please?
Okay, I'm not sure if I can do this in this class, but I'm wondering if the CFTR protein RNA is coming off of the DNA, so it's happening in the RNA and the little strand that comes off, and then it's expelled, and that's how we get the CFTR.
Yeah. Let me go back. Okay, that's so what he was showing you is this process, which is summarized in this drawing. So on the left here, you can see inside the nucleus is chromosome 7. That's where the CFTR gene is encoded. Well, when it's transcribed to mRNA, that occurs in the nucleus, but the DNA stays in the nucleus, but the mRNA can go out through a pore, and it goes immediately into this endoplasmic reticulum, this series of membranes, and that's where the ribosomes translate it into the CFTR protein. And that's what you see illustrated here. This complicated series of loops is the CFTR protein. And this is where the, it says chaperone here, that means a sugar, a simple core sugar is added. And then in a vesicle, the CFTR protein is transferred to the Golgi apparatus, where there are more sugars attached to it. These are the complex sugars. And then it goes from vesicle to vesicle until finally it's transferred into the plasma membrane. And all of this is critical for you, because what you're seeing here is what happens in wild type if there are no mutations. Now, if there's a mutation in the gene, this process is going to be affected at some point. So certain mutations cause no protein at all to be produced. Some of them end up being trapped in one of these vesicles and can't go further. And others get to the membrane, but they're defective. They can't transport chloride ions effectively. So that's what I took some screenshots from the video to try to illustrate, to try to capture in a still form what he was saying, so that you start with the gene, and he drew it all out here, talked about introns and exons, and then talked about the Golgi and the ribosomes translating the mRNA into protein, the CFTR protein, which gets decorated with sugars. And then if you go over here to the right, the fully active glycosylated protein gets put into the membrane. And he went on to talk about the recycling of it. We don't have to be concerned about that now. We want to know just what happens when it gets to the plasma membrane. Now, does that address the question that you were asking?
Yes, it does. Thank you.
Sure, Professor. I just have a quick question.
So the drawing in like in the box, the drawing that's in the box, it's well, it's explaining everything that was in the picture in the previous picture that you showed us. But the bottom drawing that she did or he did is basically the entire thing is just explaining what's on that little like thing, like is it called ribosome? It's the ribosome, right?
You want me to go back to here? The ribosomes are in the endoplasmic reticulum. These little balls here, little brown balls, are the ribosomes. And the long chain is a messenger RNA. So the ribosomes are translating the message, the code in the mRNA, translating it into protein. And that occurs in the endoplasmic reticulum. And from there, the transport continues to the Golgi, through several vesicles, and finally it gets to the membrane. And if there are no mutations, it's an active protein. It transports chloride ions effectively. And so what you're going to have is a mutation. And what you have to do is figure out where, what the effect of your mutation is on this process, going from the gene in the chromosome to the membrane and to the protein in the membrane. Is that what you're talking about?
Yes, thank you.
Professor.
Uh-huh.
I'm Elizabeth Ortiz Martinez. At one point in the video, he said something about pre-mRNA and mature mRNA, but I didn't catch what that meant. Like, what is the difference?
Okay, let's go back to here. So he's talking about splicing. So imagine that this is the CFTR gene with exons and introns. When it's transcribed, you get this pre-RNA. So the pre-RNA still has the introns in it. Then it's when the introns are spliced out, you now get what he called the mature mRNA. So that's nothing but exons joined end to end.
Oh, okay.
And real quick, does it take out the introns? Because like you were saying, those are, there's nothing in them, like it's like junk, or.
Yeah, some people refer to it as junk. It really isn't, and I'll talk about later why.
Yeah.
It's not. And one of the, well, in a nutshell, most of this junk seems to be involved in gene regulation. So it doesn't encode enzymes, but it does regulate their production.
Yeah, well, that's what I kind of figure when they always say, oh, we have all this junk DNA. Well, I'm like, if we were born with it, it probably has, some use.
Well, think about it in terms of evolution. Why would something that wasn't useful be maintained over millions of years of evolution?
Yeah,
This
good point.
doesn't make sense. So there has to be functions for all the DNA that each of our cells replicates all of this DNA every time a cell divides, which takes a huge amount of machinery and a huge amount of energy. And that just wouldn't be happening if it wasn't useful. So the idea that it's junk, it's really a misnomer. It shouldn't be called junk.
Yeah, I got you.
Okay, so let's go back to
I have a
here.
quick, I got a question about this as well.
Uh-huh.
And I'm just gonna kind of go over it again. So you start the chromosome and it goes into the DNA, it turns into the mRNA, and it goes into the reticuli.
Endoplasmic reticulum. Yeah.
Yes. And then the ribosomes essentially decodes the mRNA and it turns it into the protein, right?
Yes.
Now, between 5 and 6, what exactly is that protein doing? Or is it just leaving that reticuli into the Golgi apparatus? Is it picking up the extra sugars like he was saying, or what exactly is that area?
It picks up at about position 6, it will pick up the core sugars, the simple
Okay.
linear sugars. And then in the Golgi, the complex sugars are added.
Okay, got you.
Okay, and this we've gone over. Now, I just wanted you to have a clear picture in your mind of a core sugar is just a linear array of monomers, in this case, glucose. A complex sugar is branched. So now that we've talked about CFTR and how it's made, let's talk about mutations. The definition of a mutation is any change in the nucleotide sequence of DNA. It may result in a change in the amino acid sequence.
Gabriel,
Yeah,
muy poquita.
is there a question? Guess not. So it may change the amino acid sequence or it may not. And if it changes it, the protein may then function incorrectly. Mutations are the basis for permanent variation in organisms and provide the raw material for natural selection to choose the fittest. Mutations cause genetic diseases in humans and in other organisms as well. And cystic fibrosis is a good example of a genetic disease. It is strictly a genetic disease. You do not get cystic fibrosis unless you have a particular mutation in the CFTR gene. So what causes mutations? Well, we spent a lot of time talking about DNA replication. It's very complicated, a lot of enzymes involved. And during the copying process, mistakes can happen. And a mistake is a mutation. There are chemicals that damage DNA and cause mutations. Radiation, I'll show you an example of radiation damage to DNA. And if we have time today, I want to talk about CRISPR, the genome editing tool, which I suspect you've all heard about. And I'd like to describe it to you in some detail because it's revolutionizing biology. So here's an example of a mutation caused by UV radiation. In other words, sunlight. Sunlight could cause this. If you go out, sun hits your skin, this could happen. So if you look in the upper left, you have a DNA strand with 2 thymine residues adjacent to each other. Now, UV radiation strikes these 2 thymines and they fuse to form this structure and form what is called a pyrimidine dimer or a thymine dimer. Well, what are pyrimidines and what are purines? In the lower left, in the red box, DNA has 4 bases. Adenine and guanine are purines. Cytosine and thymine are pyrimidines. And you can see them next door here that A's and T's bind. So it's a pyrimidine binding to a purine. G's and C's are hydrogen bonded. Again, it's a pyrimidine and a purine. Now, so that's what a pyrimidine dimer is. And what you see on the right is 2 thymines. UV light strikes, causes a thymine or pyrimidine dimer. And now the DNA has to be replicated. So when DNA polymerase comes along replicating the DNA, it comes to a thymine dimer, which has caused what is known as a kink, a bend in the DNA. And the enzyme has to get past that. And when it does, it very likely will make a mistake. So a mutation will happen at the position of the kink. So UV light causes a thymine dimer. Thymine dimer causes a kink. The kink causes DNA polymerase to make a mistake, and that is a mutation. So now I want to go through some examples to illustrate the effects of mutations. So we're going to start here with an original DNA strand, and a mutation occurs. So this T is changed to an A. What is the effect on the peptide? This is something for you to do. First, you have to transcribe this mutation to mRNA, and then you have to use a codon table to translate it. So we'll do that. Here's another example where this G in the original strand is changed to an A. What is the effect on the peptide? Well, I've worked this out for you, but you should do it yourself because this will surely come up again later on tests. So if you look at the top here, this is the normal wild type sequence with no mutation. You have the sequence of the DNA, the corresponding sequence of the RNA, you use a codon table, and you get the amino acid sequence, a peptide, and this is wild type. Now, if we go to the mutant 1 sequence, mutant 1, this T becomes an A. So the mutant 1 sequence, this T here becomes an A. When that's translated, the A would be a T, but this is RNA, so it's a U. And so when you translate that, we now have, we still have a serine. And let's just go back and look to see why. So the original sequence was UCA. Now it's UCU. So UCA is serine. UCU is still serine. So this is known then as a silent mutation, because even though it is a mutation, it's a change in the DNA sequence, it doesn't change the peptide sequence. Second.
I have a question.
Yeah.
For the normal one that's DNA to RNA, doesn't thiamine translate into uracil when it's RNA, or why is it
Yes.
every time there's an A, there's a U instead?
Always. You know, if it's RNA, it's never a T, it's always a U.
Well, so in the example, I don't know if it's translating this DNA in the top, but why is it TA but then AU? Shouldn't it be TU and then AT? Then the one above it is what I'm looking at. See how it goes TAC in the very first 3? Well, shouldn't it be T? Well, it's TAC, but then below it, it should be UTG, right?
No, because that's RNA. See, this is RNA. And so the A, you're right, if it were DNA, A would become a T when it's copied, but it's RNA, so it's not a T, it's a U.
Oh, okay.
First codon is a UG.
Okay.
And so what I'm illustrating here is a silent mutation. It's a mutation, but it doesn't change anything because of the redundancy in the codon usage. UCA and UCU both code for serine. So there's no change. Now, if we go to the mutant 2 sequence, this is mutant 2, where this G becomes an A. So this G in the wild type now becomes an A. And when you transcribe that to RNA, we now have a UUA codon instead of a UCA codon. So let's look at the codon table for UUA. It was so UUA is leucine. And so now we have not only a mutation in the DNA, we have a change in the amino acid sequence. And this is called a missense mutation because it's a different peptide. So I'm making a distinction here between a silent mutation and a missense mutation, which has a consequence, a physiological consequence.
Professor, I have a question.
Yeah.
A silent mutation on a person, we wouldn't really tell, right? And then
A silent mutation, you'd see no effect in the phenotype.
and then the mutant mutation, we would tell, right? Like
Well, you may or may not. If the change in amino acid in the peptide is consequential, that is, if it changes the nature of the protein and interferes with its function, then you'd see it. But just because there's an amino acid change doesn't necessarily mean that there is a physiological effect. It may or may not have an effect.
all right, thank you.
Excuse me, I have a question.
Uh-huh.
Why now the DNA up there is G, like, and then comes to A, while I was saying like it's only to G and C together?
Which sequence are you looking at?
Like on the top, it's AGT, like the second one. And the mutation 2 sequence, it's like AAT, not like C. Why it's not C and A?
That's just arbitrary for illustrating the effect. So arbitrarily, I changed this G to an A. So supposing that it's a copying error, that when the DNA polymerase was copying this DNA, it, instead of putting a G in here, it mistakenly put in an A. So now we have a mutation. And when that's transcribed into RNA, the codon now, instead of being UCA, it's UUA. And that changes the amino acid from serine to leucine.
Professor, what would happen if there's like multiple mutations in the DNA?
You get multiple changes. So that's a good point. You can have mutations, many in a particular gene, in a particular DNA sequence. You can have many mutations. And the more mutations you have, the more consequences you're going to have in terms of the protein that's made. And so that brings me to the next point, which is types of mutation. So this is kind of a busy slide. This table on the right, table 14.1, is a set of point mutations. That's just one type of mutation. Point mutation is one base pair change. So in this table, all the mutations are just one base pair. Another type of mutation is called a duplication. And that's where a piece of DNA is copied and repeated exactly. Then there's a deletion, where a piece of DNA is lost. There's an inversion, where the DNA is just flipped over. An insertion, a new piece of DNA is introduced. A translocation, two pieces of DNA on different chromosomes, usually, trade places. So you should know all of these. And to help you visualize them, I put in this set of illustrations. So a duplication, here, this is the same chromosome. On the left, you can see the orange bands, and they're repeated again in the chromosome on the right, but then copied and repeated again. So it's exactly the same sequence of DNA repeated. That is a duplication. A deletion is the loss of one or more base pairs. And here we're talking about chromosome 4, before on the left, and after on the right. So the piece that's going to be deleted are these 4 bands, 3 blue, actually just the 3 blue bands are deleted, so that you end up with the chromosome now with this blue band here is retained, and the purple bands, 3 purple bands are retained, but what's in between them, these 3 blue bands, are lost. So that's a deletion. An inversion is just flipping a piece of DNA over. So the original chromosome is, the sequence is green, orange, blue. After the inversion, it's green, blue, orange. The different colors representing DNA sequences. And on the right is an example of a translocation. In this case, it's reciprocal in that 2 chromosomes are involved, chromosome 4 and chromosome 20. So chromosome 4 has this set of green bands at one end. Chromosome 20 has these red bands at one end, and they trade places, so that the new chromosome 4 is missing its set of green bands and it's replaced by the red bands. And in chromosome 20, its red bands are replaced by the green bands from chromosome 4. So it's just a reciprocal change of sequences, exchange of sequences. And finally, we have insertions. An insertion can be as little as 1 base pair, or it can be 3 in this case. It could be 1,000. It could be many thousands. Insertions can be anywhere from 1 base pair to kilobases. So for purposes of understanding the effect of an insertion, consider this one, insertion of 1 base pair will cause a frame shift. And a frame shift is important because all the amino acids downstream from that insertion are changed, because it changes the codons. So the normal is a CCC, now it becomes TCC. The normal would be ACT, now it's CAC. So instead of the amino acid sequence being glutamine, proline, threonine, it's now glutamine, serine, histidine, completely different. And as you go on, every amino acid would be different from normal. Now, if you inserted an entirely a complete codon, it would just add an amino acid. So TTT is the codon for phenylalanine. If you insert a TTT between this G and this C, it would result in a protein which has glutamine and now phenylalanine, but then the original sequence continues, proline and threonine. So let's see. So I show you that to illustrate, help you visualize these different types of mutation. Then what are the effects of point mutations? A silent mutation, of course, has no effect. A missense mutation is a one base pair change, alters the protein. A frame shift alters everything downstream of the point mutation. A nonsense mutation introduces a stop codon. And these are all illustrated here and to the right. And I'll describe this, and then I'm going to have to finish because we're out of time. Okay, the wild type is at the top. Mutation is none. You can see the DNA sequence, the amino acid sequence. Now here's a base substitution where this G becomes a C. But it's a silent mutation because the codon, the two codons code for the same amino acid. Now the next one down is a missense mutation. So this G in wild type becomes a C, and that changes the amino acid sequence from alanine to proline. But then the rest of the polypeptide is the same. A nonsense mutation changes this A to a T. So TAA is stopped. So the protein is truncated, it stops prematurely, and in most cases, this would be non-functional. And then at the bottom here, a one base pair change can cause a frame shift. So this C becomes an A, and that shifts the frame such that instead of having a proline here, we have a threonine, and then everything downstream of that is new, is changed. So instead of pro-lyse glue 3, it's 3-glue ARG-S. So any questions about that, about point mutations? This is all about point mutations. And what I want to emphasize is, first of all, that point mutations are very important. In fact, most of the mutations in the CFTR protein are point mutations. But there are all these other different types of mutation that you should know about and be able to visualize. I'm going to have to stop there, but remind you that I'd like you to think about this information that we've gone over today and respond to the assignment that I posted, and send me a concern that you have that you'd like to discuss on Monday. And we'll do that first thing. I'll try to consolidate. I'm sure some of you will have the same concerns. I'll try to consolidate them so that we have just a limited number of issues that we need to focus on Monday. Anything else? Okay.
You know what? I don't know if this will help anybody else, so they don't really have to stay, but when we were talking about transcoding that DNA to RNA, I think I'm still a little confused on it.
Okay, we'll talk about it on Monday. So send me that as a question, and I'll include it into a topic that we'll talk about on
Okay,
Monday.
that sounds good. Thank you. Have a good day.
Okay,
Have a nice day.
okay. See you all on
Thank you, Professor.
Thank you.
Monday.
Thank you,
Thank you.
Professor.
Bye,
Okay,
everyone.
Thank you.
all right.
Professor, I have a question.
Uh,
I think can you add me on a group for the cystic fibrosis project? Because I think I'm not in a group.
I haven't assigned groups yet.
Oh, okay.
I will do
Oh.
that. We will have a big discussion about the project. And at that time, or after we do that, I will assign people to groups.
Oh, okay. Okay, that's perfect. Thank you.
Uh-huh.
Bye. Have a great day.
Bye.
Professor, I forgot to tell you my name when I spoke earlier today, and I just want to make sure you wrote my name down.
Okay. All right. What's your question?
Oh, I don't have a question. Earlier I asked about the where the CFTR happened, if it happened after, like in the RNA strand earlier, but I forgot to say my name before I asked you the question, so I just want to make sure that you wrote me down when I spoke.
Oh, okay.
Yes, that's Alicia Carroll.
Yeah, okay. I noted that down. Okay.
Okay, thank you. Have a great day.
All right. Take care. Bye.
Bye-bye.